So welcome back again from the break, whether you stepped away or whether you clicked on through. Let's talk about the concept check questions and get these answered so we can talk about the last part of the chapter. How does cytokinesis in animal and plant cells differ and why? Remember that in cytokinesis in animal cells, it's simply a case of pinching down through the center of the cell and pinching them into two separate daughter cells. In plant cells, you have the formation of the cell plate, the lining up of the vesicles that are going to give rise to the formation of the cell plate. As it expands, it forms the cell wall that ultimately splits the two daughter cells into half, or splits the parent cell into the two daughter cells. Why the difference? Because plant cells have a cell wall and they can't survive without a fully developed, fully functional cell wall. So the cell wall, or developing the new cell wall, is a very good way to carry out cytokinesis. In animal cells, doesn't matter. There's no cell wall. They can simply pinch in half. During which stages of the cell cycle does a chromosome consist of two identical chromatids? Well, the two sister chromatids form up when the DNA molecule is replicated. So we would say that the first time that we're going to see those two sister chromatids is at the end of S. We would have the two sister chromatids. Those would continue as two sister chromatids during all of G2, then on into prophase and prometaphase and metaphase, and the two sister chromatids would ultimately separate from each other in anaphase of mitosis. So if we wanted to list them, that would be the listing. So it would be from the end of S through G2 through prophase, metaphase, or prophase, prometaphase, and metaphase, and they would separate in anaphase. Or we could say from the end of S through to the beginning of anaphase, however you want to express it. But that would be where we would see each chromosome consisting of two identical sister chromatids. So one of the things that we need to recognize is that cell division does not occur arbitrarily, nor would we want it to. And for the most part, the vast majority of cells in our body, once we achieve our full adult size, stop dividing and go into what we might refer to as a maintenance phase, and only start dividing again if it's necessary for cell replacement or repair of some damage that might have been done. Otherwise, the vast majority of cells are in this maintenance mode. And in the case of some cells, most notably the nerve cells of the spine, they are permanently in that maintenance mode, which is the reason why spinal damage is so devastating, because unlike other cells where damage might occur, those cells could be replaced. In the spine, if those nerve cells are damaged, they can't be replaced. They are locked in a non-division state, or at least that is the general consensus. Now, there is experimental work that suggests that we may be able to change that, because we know more and more about what regulates cell division and what causes cells to enter that maintenance mode and how we might get them out of it again. But that lies beyond the scope of this class. But again, if your intention is to take the General Biology 2 class, we do get into talking about that when we talk about embryological development, because it features in that discussion. So we do know a lot more in our ability to be able to encourage cells to divide in circumstances where we might not expect them to divide, and particularly to get them to take on different roles that lay outside of their normal role, is quite spectacular. But as I said, it lies outside of the scope of this class for now. But again, if your intention is to take the General Biology 2 class, you'll learn more about that when we do the section on embryological development. But the point here is, it appears that cell division must be under tight control, and there must be a mechanism for controlling that. And we can demonstrate that fairly easily. So here is a diagram that illustrates two separate experiments. Now I want to make a clarification here. These cells do not survive outside of this experimentation. So the goal here is not to make some sort of Franken-cell. It is, however, to see the effect of fusing cells together that are in different stages of the cell cycle. The first one I don't think is a particularly gee whiz type of experiment, but the second one most certainly is. So let's talk about the two of them. So here we have two cells. One cell is in the S phase, so it's busily replicating its DNA at the moment. The second cell is in G1, so it has technically just finished the division process. It would be going into the S phase at some point, but probably not right now. But if you fuse these cells together, and that's relatively easy to do, we've talked about how to do this previously, the cell that was in G1 immediately transitions into S phase. And it begins duplicating the chromosomes. So a cell that was probably going to lag in G1 for a while immediately enters S without hesitation, all because it's been exposed to the cytoplasm of a cell already in S. Perhaps not so much of a gee whiz, but nonetheless, it illustrates that there must be something in this cell that is affecting the rate at which this cell enters S. But I think the next one really reinforces the fact that there are some very powerful molecules present in the cytoplasm of cells. Here we have a cell in G1. It's just finished the division process. It will not enter mitosis again until it's passed through S and passed through G2, and then we might expect it to enter mitosis, assuming that it's given permission to go in that direction. That's not a given, as you'll see. Here we have a cell in mitosis, so it's actively dividing. In fact, we might have presumed by this diagram that it's in prophase. Look at what happens when you fuse the two cells together. Immediately, the cell that was in G1 enters something that looks like mitosis, and it actually looks like prophase. Now, of course, it hasn't duplicated its DNA, but that doesn't stop it from doing, or at least attempting to getting into prophase and starting prophase. The spindle starts to form, the nuclear envelope starts to break down, all things that we associate with prophase and prometaphase. Yet it has not duplicated its chromosomes. It's not even supposed to be ready to get into mitosis. Yet just by being exposed to the cytoplasm of a cell in mitosis, it immediately enters mitosis. That tells us that there must be some components in the cytoplasm, probably proteins, we now know that they are proteins by and large, that are very, very potent, very powerful. They can override the normal programming of a cell and change the way this cell, a cell, progresses through the cell cycle. These are very, very powerful molecules, and we call them growth factors. So I think I'll put, I'm going to put a note here. So these molecules are called growth factors, and these are very, very powerful molecules. Now we know about the control of the cell cycle because we made a commitment back in the early 1970s to figure out how to defeat cancer. And of course, what you may hopefully already know about cancer cells is that these are cells that are growing and dividing in an unchecked fashion. In other words, they're defying whatever it is that regulates the growth and division of cells. And so in order to try and figure out how to defeat cancer, you first have to figure out what it is that causes cells to grow and divide in the first place. And so everything that I'm about to explain to you about the regulation of cell division has come out of an understanding of what it is that makes a cell cancerous and how we might be able to either prevent that or kill off cells that have started down that road. So the first thing to recognize is that there are 3 key points in the cell cycle where regulation is imposed on cell division. The first one and the most important one is what is referred to as the G1 checkpoint. This is the checkpoint at which it is decided whether the cell will continue on to divide or not. And so we might put this as the divide. That's not going to look very nice. I really struggle to write way up here on the, it's because it's, you may not realize this, that the tablet's on an angle and I'm trying to reach up here and brace myself without pressing too hard on the tablet. So this is the divide or not divide. So that's the question that's being asked here. So is the cell going to be given permission to divide or not divide at this point? Down here at the second checkpoint, we're going to talk a little bit more about the G2 checkpoint. Here it's mitosis or not mitosis. So here, basically at this checkpoint, an internal self-check is run to determine whether the cell is ready to begin mitosis and to undergo mitosis. Is everything in place? Are all the components in place? Have all the chromosomes been copied correctly? Is each chromosome composed of 2 sister chromatids? Is everything there ready to go? If the answer is yes, after that self-check has been run, then the cell is allowed to enter mitosis. There's one more checkpoint called the M checkpoint. This is the metaphase checkpoint. And here it's anaphase or not anaphase. And again, there is an internal checkpoint or internal self-check run. And basically what's being looked for here is that each of the sister chromatids is connected to the spindle via a kinetochore, so that each chromatid is connected to one end of the spindle, that the other sister chromatid is connected to the other end of the spindle, that each of them has microtubules to attach to the kinetochore to successfully separate the 2 sister chromatids, and that all of the chromosomes are in this condition. And if the self-check says yes, all of that is okay, then they're allowed to pass through the M checkpoint and enter anaphase and complete mitosis. If they fail the G2 checkpoint and the M checkpoint, then there are mechanisms in place that take those cells and kill them off, by virtue of the fact that these cells can no longer be allowed to divide because they're not in a healthy state to divide. If it's possible for an internal repair, if it's just a simple error, then that can potentially take place. But if it's major internal damage, that means these cells simply cannot be allowed to divide because they're not healthy enough to be allowed to divide, then there are processes in place, and the major process is called apoptosis or programmed cell death. Or you could say suicide. The cells are actually programmed to die off. And there is a genetic control over this. And the genetic control is one of the things that we'll talk about, because it's something that we've learnt more about in studying cancer, the fact that cancer cells seem to get round, or have figured out ways to get around, this programmed cell death, because you would think, seeing as how a cancer cell is, or a tumor cell is going to become potentially dangerous to the organism, it's an unhealthy cell, that apoptosis would kill it off. And under normal circumstances, a cell that is tending in that direction would get killed off via apoptosis. But as you'll see, tumor cells and cancer cells specifically get around this whole control mechanism. So the growth factors are what control the passage of the cell past these checkpoints. So as I said, the chief checkpoint is the G1 checkpoint. So once a cell gets to the G1 checkpoint, if it is not able to get through the G1 checkpoint, rather than dying, it enters a new stage which we've defined as G0, and G0 is this maintenance stage. Remember, the vast majority of cells are in this maintenance stage. So this is where most cells wind up. They simply lack permission to continue on, because once you get past G1, there is this essential commitment to getting through, to begin getting ready for division. So this is kind of important. If you get through G1, you're essentially being given permission to go ahead and get ready to divide. Of course, you'll be checked internally twice to make sure that it is still okay for you to divide, but the G1 checkpoint is where that basic decision, divide or not divide, is made. If the decision is not divide, then you enter maintenance mode, the G0 mode. Once you enter, get past the G1 checkpoint, then you go through S, you go through G2, and you run up against the G2 checkpoint. Again, remember here, it's all about, are you in a healthy condition to begin mitosis? Are all the chromosomes copied? Do all the chromosomes consist of 2 sister chromatids? Are all the components in place to allow mitosis to begin successfully? Yes, no. If the answer is yes, move through the G2 checkpoint and enter on into mitosis. And then you have the M checkpoint, end of metaphase, beginning of anaphase. Are all the chromosomes connected to the microtubule, to the spindle via the kinetochore microtubules? Is each sister chromatid connected to one end of the spindle? Is everything lined up in the center? All that checkpoint at metaphase. If the answer is yes, then it continues on through anaphase, and mitosis completes. So you can see that these checkpoints are really important, and the presence of growth factors at each of these checkpoints is what determines whether the cells pass through them or not. So what we're going to do is take a look at the key growth factor at the G2 checkpoint. A protein called MPF, or a growth factor called MPF, which is typically referred to as maturation promoting factor. But I noticed that the textbook has taken on calling it something that I've called it for a little while now, and that's mitosis promoting factor. Because that's really what this is all about. This is the growth factor that needs to be present at the G2 checkpoint in order to allow the cell to begin mitosis. So it's really promoting mitosis. Now, it turns out that these internal growth factors, because there are 2 different types of growth factors, internal and external. We'll talk about the external shortly. But the internal growth factors consist of 2 components. A group of proteins called cyclins, which vary in concentration according to which part of the cell cycle you're in. So their levels go up and down, hence the term cycling. And then a series of enzymes called kinases. Now, a kinase adds a phosphate to something. So kinase enzymes are activators of other enzymes and other proteins. But these kinases do not function independently. They're referred to as cyclin-dependent kinases, or CDKs, meaning they will only work in the presence of cyclin, or to be more specific, in the presence of their particular cyclin. Because it turns out that there are a series of these kinases and there are a series of cyclins, and each kinase is dependent on the presence of a particular cyclin. So we know of about 8 different cyclins now. Some proteins have been referred to as cyclins and then subsequently been removed from the list. So if you take an advanced biology class, you may encounter the cyclins again, and you may encounter the names of some of those cyclins. And you'll find that they're given numbers. And so then some of the numbers actually reflect the fact that we've thought in the past that some proteins were cyclins, but now we don't think that they are, but the number was already taken, so we can't reuse the number, so we've had to use extra numbers to take this into account. That being said, we're going to talk about a particular cyclin-dependent kinase and its cyclin, and it's one of the first that was ever discovered, and it forms this maturation promoting factor, or as I like to refer to it, mitosis promoting factor, because that more accurately reflects what it does. So let's take a look at the diagram that illustrates how this works. So when we look first for MPF activity, we notice that it shows a cyclical pattern. It begins right as mitosis begins. It peaks in the middle of mitosis. In fact, it peaks right around metaphase. And then it drops away very rapidly and reaches 0 as mitosis is finishing. Something that we might logically expect to see happening for a factor that is promoting mitosis. We would expect its activity to begin with mitosis, that it increases as it's promoting mitosis, and then as we start to reach the end of mitosis, to see its activity decrease again. So not surprisingly, MPF activity correlates very nicely with mitosis, beginning when mitosis begins, peaking in the middle of mitosis, ending as mitosis ends, something that we would expect. But when we look at the 2 components of MPF, the cyclin and the cyclin-dependent kinase, what we notice is that the kinase is present at a constant level. It neither rises or falls. It's present all the time, even though MPF activity rises and falls in correlation with mitosis. So it can't be the cyclin-dependent kinase, it can't be the CDK, the kinase, that's the determinant of activity. When we look at the cyclin concentration, all is revealed, because we see that the cyclin concentration goes up and down in concert with MPF activity. So it's the appearance and disappearance of cyclin that determines the presence and disappearance of MPF activity as a promoter of mitosis. It's a little bit more complicated than that, because as you take a closer look at the concentration of cyclin, what you notice is that it actually starts to be produced back around the middle towards the end of S, but MPF activity doesn't begin until the end of G2, beginning of mitosis. So why the disconnect between the appearance of cyclin and the appearance of MPF? Well, the answer is because cyclin does not start to bind to the CDK until its level reaches a threshold level. And then MPF activity does not begin until the amount of MPF reaches a threshold level. So just because you've got cyclin present and just because the cyclin is now binding to the kinase does not automatically mean that the MPF activity is going to begin or that mitosis is going to begin. The MPF concentration has to reach a certain threshold level before it will trigger the beginning of mitosis. And it's that rise of MPF activity that constitutes the internal self-check, because remember, MPF is an activator and it's not going to start activating anything until its level reaches the threshold, until that internal self-check says, everything's good, we're ready to go. Then MPF activity rises in parallel with the increase in cyclin concentration. But the MPF activity declines again when cyclin concentration declines. And the reason why is because throughout all of this time period that cyclin is accumulating, it's specifically protected from being degraded. But at this point, when it reaches its maximum, that protection is removed and cyclin is now degraded very, very rapidly all the way back to zero. And that is what leads to the loss of MPF activity. Because in the absence of cyclin, the cyclin-dependent kinase shuts down, no more promotion of mitosis. And that happens very, very quickly. And throughout this period, where we see virtually no cyclin, the presence of cyclin is actually shut down. If any cyclin this cyclin was to appear, it would be broken down because its protection has been removed. But we know that during this period, cyclin is simply not being made anyway. Once we get towards the end of S, the protective mechanism reappears and cyclin production is turned on. It's one of the things that occurs during the S period, so that during the whole of G2, cyclin concentration is increasing. The appearance of MPF starts to appear. When both reach that threshold level, MPF activity triggers the beginning of mitosis by activating the components that are necessary for the beginning of mitosis. And all of the growth factors that appear at G1, at G2, at M, the growth factors that are necessary at each of those checkpoints function in much the same fashion. They involve this two-component enzyme system, a kinase that activates other proteins that trigger the various steps that are being promoted, and a cyclin that attaches to the kinase to turn that kinase on at a particular point in time, the G1 checkpoint, the G2 checkpoint, and the M checkpoint in the cell cycle. They all function in much the same way. These are the internal growth factors. They're very, very powerful molecules, as you've seen. They can totally overwrite the normal programming of a cell simply by being exposed to them. But there are a second set of growth factors that function from outside of the cell. So while these growth factors are allowing the cell to progress through the cell cycle, remember at G1, you have to decide whether the cell is going to be remain within the cell cycle or exit the cell cycle into G2. And also, if a cell needs to re-enter the cell cycle to exit G0 and enter G1 again, it needs to get the message to re-enter the cell cycle. So there are external growth factors that signal to the cell whether it's appropriate to be in the cell cycle and grow and divide, or whether it's appropriate to exit the cell cycle and not grow and divide, be in maintenance mode. So we can demonstrate the existence of these growth factors using a system that is associated with plugging up cuts or breakages to the circulatory system. So this is a mechanism that would kick in if you cut yourself. You know that initially you bleed for a few minutes, but eventually the blood flow stops. You see a little bit of the interstitial fluid, that's the clear fluid, otherwise known as blood plasma. But eventually you get a scab. And over time, with the scab protecting the cut, the underlying skin cells divide and heal up the cut. And eventually the scab disappears again and you've got a healed cut and there's no more cell division. That clearly indicates that when you cut yourself, a series of events must be triggered that lead to initially a scab that is going to become a semi-permanent or a temporary seal, I guess, until the skin cells can seal up the cut, and then everything shuts down again. And this is that temporary seal. The scab is the product of the blood plasma clotting factors in a group of cells called fibroblasts. Now, the fibroblasts are present all the time, but they're not going to grow until a cut is detected. And that cut is detected by a group of very small cells that are called platelets. These are actually fragments of larger cells. And the platelets are constantly circulating in the bloodstream. There are millions upon millions of them, and they are the first type of cells to turn up in the cut that recognize that the cut exists. And they are the ones that stop the loss of red blood cells. The interstitial fluid that is going to help form the scab is still there. The fibroblasts are going to contribute to the formation of the scab. But they are only going to do that because they recognize that it's time for them to start growing and dividing. And it's the platelets that provide the signal. I may have called them something else, but they're called platelets. So PDGF stands for platelet derived growth factor. So it's possible to isolate fibroblasts relatively easily from human tissue if you put them into a mixture of enzymes that break down the extracellular matrix. You can free up the cells themselves and put them into these special chambers that have been designed for growing cells. They have a flat bottom because the largest number of cells, the vast majority of cells in our body, with the exception of a few blood cells, require that they be anchored to a surface in order to be able to grow and divide. It's called anchorage dependence. And it is, in fact, one of the things that prevents cancer cells from growing, the fact that they need to be anchored to something. The exceptions are cancer cells of the blood, so leukemias and things like that. Those cells don't require an anchorage point, so they have an independent control mechanism. They don't need to be anchored to something. But all animal cells and cells in the human body respond to something called density-dependent inhibition. And the growth of cells inside these chambers illustrates that. So if you put the fibroblasts into these chambers and you give them all the nutrients that they need, but one chamber has the PDGF present, the other chamber does not, it's only within the chamber that the PDGF is present that you're going to see cell growth. The two flasks are identical. One has PDGF, the other doesn't. So that tells you that the platelet-derived growth factor, this product of the platelets that are the first cells to recognize that the cut exists, is the reason why the fibroblasts start to grow to help form the scab. It's going to be that temporary block to the cut until the skin cells can seal up the cut permanently. And so the fibroblasts start to grow. If we leave the PDGF in the solution, they'll continue to grow until they cover the bottom of the flask, and then they'll stop. And this is the density-dependent inhibition kicking in. What we believe is happening here, and the evidence is pretty good for this, is that as the cells start to touch each other, they stop producing the internal growth factors. And that eventually results in a loss of the internal growth factors once the cells are touching each other and there's no further room for them to divide into. This is this notion of density-dependent inhibition. And we know that it's that physical touching of the cells that's causing this, because if you reach in with a spatula and scrape out some of the cells and create a hole, the cells immediately start growing again. They re-enter the cell cycle, those that are on the edge of the break, and start growing until they fill in the space. Once the space is filled, they stop growing again, with one exception. If you grow tumor cells, fibroblast tumor cells, they will continue to grow irrespective of whether they have filled the available space or not, because they totally ignore the density-dependent inhibition. They've found a way around that. And so tumor cells are very specifically cells that ignore density-dependent inhibition. And this is typically why tumors, and specifically cancers, wind up killing you, because they're occupying space that ought to be occupied by healthy tissue. They're growing and forcing the healthy tissue out. They're taking up resources that normally would be used by healthy tissue. And as a consequence, they damage the local area that they're growing in, and eventually it winds up killing you. But it's not just any tumor that winds up killing you, because it turns out that there are two types of tumors. So the initial tumor is referred to as a benign tumor. So a benign tumor is one that grows in the local area where the cells first appeared. And different cancers are identified by the type of cells that are involved. So the lung cancer is lung cells, a brain cancer is brain cells, a bone cancer is bone cells, and so forth. And breast cancer is breast cells, obviously. So different types of cancers are identified by the types of cells that are involved. But a benign tumor, by and large, is unlikely to kill you. And typically, if you have a benign tumor, there is very little rush to get rid of it, so long as there is really good evidence that it's going to stay as a benign tumor. Unfortunately, for the case of some types of cancer, so lung cancer, colon cancer, skin cancer, it is very likely that the tumor is not going to remain benign for very long, because once it gets access to either the bloodstream or to the other plumbing system of the body known as the lymph system, cells from the benign tumor will break away and move to another location where they'll set up home and start growing again. And then we say that the tumor is now malignant. We say that it is metastasized. Oh, excuse me. So officially, in the medical world at least, we don't name things a cancer until the tumor has metastasized and you've got a tumor growing in another location. So if you've got lung cancer, it's probably as a result of a benign tumor moving to another location. Interestingly, very few people actually get brain cancer. Brain cells do not normally become cancerous, although it does happen. But cancer in the brain is often lung cancer that has metastasized, or a lung tumor that has metastasized, and the cells have migrated to the brain and started growing there, forming a malignant tumor inside the brain. And most lung cancers are not identified initially as a lung cancer. It's usually because it metastasizes somewhere else. Likewise with colon cancer, unfortunately, and to some degree bone cancer and skin cancer as well. So one of the things that we do know is that early detection is the key to surviving cancer. And so there are some things that we can do to make sure that we catch them while they're still in their benign stage, which unfortunately for some cancers is relatively difficult to do because of where the tumors form. But in the case of cancers like colon cancer, you can get screenings, a colonoscopy. I noticed that they've lowered the age for colonoscopy, the basic baseline colonoscopy. It used to be 50 years of age, but it's now down to 45. And my guess is they may even take it even lower, because unfortunately colon cancers are turning up in younger and younger individuals. And so as uncomfortable as a colonoscopy sounds, let me assure you it's not as bad as it seems. The actual procedure is pretty simple. They give you an anesthetic. You are asleep throughout the whole of the procedure, and the anesthetic has an amnesiac effect. It means that you have no memory of the procedure at all. You basically lose all focus, and it works really quickly. I remember I've had 3 colonoscopies right now. I'm probably likely to have another one next year, unfortunately. But they get you to count back from 100, and I don't think I've ever got past 99 or 98 and counting backwards. And then the next thing I know, I'm waking up in the recovery room, and it's half an hour to 3/4 of an hour later, and I have no memory of the time in between, which is probably a good thing, but we won't go there. But colonoscopies are pretty straightforward, and while they're there, if they find anything suspicious, they can remove it, which is a good thing. In the case of breast cancer, unfortunately, the tumors have to get to a certain size before you can physically detect them through breast exam. And by the way, guys, you have breast tissue, you can get breast cancer in case you're not aware of that. You ought to be doing breast self-exams to find any lumps that may appear, because breast cancer in males typically is more serious because males tend to not pay attention, and it gets much more advanced before it's recognized, which means that the prognosis is not good. Women are more sensitive, and therefore to the idea of breast cancer, because it's promoted, which is a good thing. Don't get me wrong, it's a good thing that it be promoted, and there are obviously ways of screening for it through mammograms. There is a lot of discussion about the value of mammograms right now. That's something that you may want to talk to with your general physician, your family doctor, just to find out what the general feeling is about the value of a regular mammogram. The point is to try and find these things fairly early on. The problem is for breast cancer, unfortunately, there are other things, most notably cysts, that can occur in the breast that resemble tumors to touch, and to some degree on a mammogram, which is just an X-ray, and can lead to a false positive, which means you have to have a biopsy that you probably otherwise wouldn't have. But by the same token, if you do have a tumor, if you find it early, you can have surgery to remove it. And again, it's taken care of right there and then. Here's where it gets bad, when the tumor expands to come in contact with the blood vessels or the lymph system, and the tumor cells get loose into these two systems. What is the lymph system? Well, it actually collects all the interstitial fluid, the fluid that leaks out of the bloodstream. That's the clear fluid that leaks when you cut yourself. That gets collected and returned back to the bloodstream again just before the blood enters the heart to keep the volume up. Scattered along the lymph system are lymph nodes, which are part of the immune system. And this is where the cells that remember how to make antibodies against different pathogens are located. So when we talk about making antibodies against the COVID virus or any other pathogen that we make antibodies against, it is primarily cells that sit in the lymph nodes that remember how to do this. And the reason why is because pathogens tend to get picked up into the lymph system as well. So it makes sense that the immune system via the lymph system should be checking for the presence of pathogens there. And cancer cells, if they get detected in the lymph system, can be taken care of by the immune system, because the immune system can identify cancer cells. And this is one of the reasons why your immune system is an important part of the puzzle in beating cancer, because cancer cells, being abnormal cells, should be identified by the immune system and taken care of. But if the cancer cells are able to disguise themselves in some fashion or avoid the immune system, that gives them obviously the opportunity to become dangerous. So this is all part of the puzzle of, part and parcel, I guess, of the puzzle of cancer. So what other things can happen? Well, as you might expect, there are genes that are responsible for identifying cells that are becoming potentially cancerous. These are referred to as tumor suppressor genes. There are also genes that are involved in the in regulating the cell division process, specifically, or primarily, those that are involved in the external growth factor mechanism that can also get altered. These are often referred to as oncogenes, which is an unfortunate name because this suggests that these are cancer genes, which is not the case. There are actually no genes in our genome whose job it is to make us sick. All genes have a normal role, and all of these genes are involved in telling cells whether it's appropriate to divide or not. They're involved in, as growth factors, in signaling whether it's appropriate for the cells to divide or not. But not surprisingly, if those genes should get mutated so that they send the wrong signal, which is what we're talking about here, then they are going to contribute to the cells changing from normal cells to pre-tumor cells to tumor cells to cancer cells. And that's basically the pathway that cells follow. And it involves not the change in a single gene, but the change in multiple genes. And it's typically thought that it requires the accumulation of about 5 or 6 gene mutations. So about 5 or 6 of these signaling proteins have to be altered in order for a cell to go from a normal healthy cell to a pre-tumor cell to a tumor cell to a pre-cancer cell to a cancer cell. And so you would have to be terribly unlucky over the course of a lifetime to accumulate all of those changes. And yet we know that that does happen. Now, some of it could be just bad luck. Some of it could be because we have inherited some of the cells or some of the genes already altered. That's why there is an inherited component or a familial component to cancer. So if you have family relatives specifically brothers and sisters that have cancer, then you have a higher than average risk of developing the same cancer because you have at least 50% of the same genes, probably more than that. If it is a parent, then your risk is not as great because you really do only share about 50% of the same genes as your parent that has cancer. If it's an aunt or uncle, the risk is increased, but nowhere near as high as it is with a parent or with a brother or sister. So the more distantly removed the family member is from you, the less is the likelihood of an increased risk. But nonetheless, we have to bear in mind that that is the potential. So this is the reason why understanding the inherited side of cancer becomes important, because if you happen to inherit some of these mutations already in place, it simply means that you have to accumulate fewer mutations in order for the cancer to develop. That's what we mean by an increased risk. You have to accumulate fewer changes in order for the cancer to develop. What we also are learning is that every person's version of a cancer is unique to themselves, that there are a number of different types of changes that can lead to the same type of cancer. So there are several different types of pancreatic cancer. There are several different types of skin cancer. There are several different types of colon cancer. All of those are different because of the different types of cells that they affect. But even two people who have exactly the same version of skin cancer or the same version of pancreatic cancer may not have exactly the same genetic alterations. And this is the reason why personalized treatment and personalized medicine is likely to become more and more common and more important in addressing some of these sorts of diseases where genes have a role, and particularly in the case of cancer. So one of the things that I would have played with if we were doing this in the classroom is a set of cards that I got from the Howard Hughes Medical Institute that outlines the sorts of genetic changes that are associated with different types of cancer. And many of those cards duplicate different types of cancers, but identify a different set of mutations involved in two different types of the same cancer. And so even being diagnosed with the same cancer as someone else may not necessarily mean that you're going to be treated the same, because it turns out that chemotherapy and chemotherapy drugs affect different types of cancer and different mutations that are involved in different types of cancer in different ways. And so it would not make sense to utilize a drug on your cancer if that drug can be shown to be unaffected or non-effective, ineffective is the appropriate term, I guess, for your particular type of cancer because of the mutations that led to your type of cancer. So there is a system that is being experimented with. I believe that would be the appropriate way of talking about it now, where cells from a particular cancer are being implanted under the skin of a type of laboratory mouse that has been developed specifically so that it will grow cancers derived from humans. It has no immune system. Its immune system has been completely, knocked out, so that a cancer from a human can grow in the body of this mouse. And so mice are created that are growing a particular cancer, and then that cancer is treated in the mouse with different chemotherapy drugs to see which of the chemotherapy drugs are going to be the most effective against that cancer. And then once you can identify the drug or combination of drugs that is most effective against that cancer, that's what you use to treat the patient. because now you know from the basis of this experimentation that this particular combination of drugs is very effective against that cancer, and other combination of drugs wouldn't work. And so here you're starting to personalize how you treat the cancer. And because different combinations of mutations can cause exactly the same type of cancer, it's becoming really, really important and more common for these sorts of studies to be done as part of the initial treatment for a cancer. Now, if the cancer metastasizes so that the cells are now, or tumors now growing in other locations, then you may have to use not a targeted type of chemotherapy, but a more systemic type of chemotherapy, meaning that it circulates throughout the body and kills the cells wherever they're located. So it is possible to target chemotherapy to a particular location of a cancer, but once metastasis occurs and the cancer cells are now circulating around in the body, you have to now use what is called a systemic approach, which targets the whole body, the whole system. And so that's what radiation therapy will do. So ironically, radiation can not only give rise to cancer by causing mutations that increase the likelihood of a cell becoming cancerous, but used in the right way, it can also damage the DNA of a cancer cell so severely that the cell simply can't survive. Now, it shouldn't come as too much of a surprise that chemotherapy drugs target components of mitosis and a favorite one is the spindle and the formation of the spindle. And in the case of radiation, well, let me put it here, or rapidly dividing cells, because cancer cells divide more rapidly than normal somatic cells, and likewise with radiation. And the reason why that's important is because, as we'll discover in Chapter 17, there are mechanisms in place to repair DNA damage. And under normal circumstances, your cells are pretty good at repairing DNA damage that occurs, quote unquote, naturally or normally. It's when that system gets overloaded with DNA damage that the system breaks down. So if you've got a cancer cell that's already dividing reasonably rapidly, if you can damage its DNA severely, the repair mechanisms simply can't handle that, and the cell will die as a consequence because it'll be programmed to die. Now, remember I talked about apoptosis and tumor cells being programmed to die as a consequence of that? Well, this is one of the tumor suppressor genes. So a tumor suppressor gene called P56 is the one that is most commonly associated with apoptosis. And this is a gene that very frequently gets mutated in different types of cancer. Because again, if the cells are rapidly dividing for reasons which we will talk a little bit about when we get to Chapter 16, they should really be veered off the normal pathway into the pathway of apoptosis. The P56 tumor suppressor gene ought to deal with them and send them off into apoptosis. But because the P56 gene has been mutated, it no longer is effective And it allows the cells to basically become immortal and grow and divide without being damaged by apoptosis. So that's why the chemotherapy and the radiation therapy targets both rapidly dividing cells and also targets the mechanisms of cell division, because that's essentially the only way of dealing with these cells, because the normal mechanisms for preventing them from dividing simply are no longer effective. They've found ways around that. Now, it's also the targeting of rapidly dividing cells that leads to issues for people who are being treated. Two common problems are anemia, loss of red blood cells, because we produce lots of red blood cells every day. We turn over all our red blood cells every 3 or 4 weeks. And so we're obviously producing a lot of red blood cells. And those are all being produced in the bone marrow in the large bones of our body. So the large bones of our legs, our arms, and the sternum, that central bone in the center of your rib cage, is where the bone marrow is located, and that's where your red blood cells are coming from. But it's also where the cells of your immune system are coming from, the so-called lymphocytes, the white blood cells. And so those get damaged. And so it's not unusual for people who are being treated for cancer to also be susceptible to infections that most of us would not even worry about, because our immune systems are active. It's also the reason why in treating blood cancers, so leukemias and the like, one of the treatments is to actually remove the bone marrow by radiation treatment to kill off all the cells in the bone marrow, and it can be done chemically as well, and then do a bone marrow transplant. The problem with that is that while it cures the cancer, so long as you can find a compatible donor, and if you're not a donor, please think about becoming one, because you could save someone's life simply by donating a little bit of bone marrow to them and allowing them to get a healthy bone marrow system. And this is very important amongst certain ethnic groups where there are markers. So what we're looking at when we do tissue transplant is markers on the surface of cells, and different ethnic groups have different combinations of markers that in some cases become really important for these sorts of tissue transplants. So in some cases, we need to expand the donor database. So if you haven't considered becoming a blood donor or a bone marrow donor, please consider doing so, because there is always a demand for whole blood, red blood cells, platelets, and for the treatment of some cancers, bone marrow. So please consider becoming a donor if you are not already. Well, I'm trying to remember where I was going with all of this. So if you do a tissue transplant, one of the things that becomes an issue is the idea of rejection, where the tissue is not accepted by the body because of incompatibility or slight incompatibility between immune systems. And so they have to take what are referred to as immune suppressive drugs. And that becomes an issue because that means that the immune system is not as active as it might normally be. And again, it means that they are more susceptible to infections that people with healthy immune systems would simply shrug off because the immune system would take care of it. And this is one of the things that we've encountered with the COVID vaccine. It's been one of those things where we've had to be really careful about giving vaccines to people who have suppressed immune system. But fortunately, the studies have been done. We now consider the vaccines to be safe for people who have suppressed immune systems or otherwise compromised immune systems. So if you have any hesitancy by all means, talk to your physician, ask them about whether it's appropriate for you to be vaccinated or whether you need to wait for a little while. But hopefully at some point, everyone will be able to be vaccinated, irrespective of their immune system status at that point. So one more concept check. That was actually the last slide in the sequence, but one more concept check. So we're really not going to take a break. Just going to do a concept check here. So how does MPF allow a cell to pass the G2 checkpoint and begin mitosis? By accumulating to a level that allows it to activate the necessary components for the beginning of mitosis. What allows it to rise to that level? By an increase in the amount of cyclin to the point where it starts to bind to the cyclin-dependent kinase, which triggers the onset of MPF activity. And once enough cyclin is bound to enough of the kinases to produce enough active kinases, then you reach the threshold that triggers the beginning of mitosis. What differentiates a benign tumor from a malignant tumor? Remember, benign tumors are growing in the local region or local area where the tumor began. So it hasn't broken away yet. There were no other secondary tumors growing in other locations. You just got the primary tumor as a single tumor growing at its point of origin. How does a malignant tumor differ? That's when the cells break away from the primary tumor, migrate via the bloodstream or the lymph system to another location where they develop into a secondary tumor. And remember, it's the malignant or secondary tumors that are now officially titled cancer, and those are the ones that will kill you because of their growth in this new location, putting pressure on healthy tissue and taking resources away from otherwise healthy tissue. That's what winds up killing you. One more concept check question. An organism called a plasmodial slime mold consists of a huge cytoplasmic mass with many nuclei. These guys really do exist. Using your knowledge of mitosis, how can you account for this? Well, remember, mitosis is separate from cytokinesis. So if you wanted to stop the cell from dividing, but still produce a lot of nuclei, all you need to do is inhibit cytokinesis, but still allow mitosis to occur. And that's what actually happens in these plasmodial slime molds. Within the original cell that produces these, the origin cell that gives rise to this huge cytoplasmic mass, cytokinesis is specifically inhibited. So the cytoplasm never divides, but mitosis continues. And so over time, you get an accumulation of many nuclei in this large cytoplasmic mass. The cell continues to grow in terms of internal volume, but it never divides into individual cells, individual daughter cells, even though it continues to produce nuclei. So that's how we can account for the occurrence of this particular organism. And there are a whole bunch of these plasmodial slime molds. And they're of real interest because of this ability to inhibit cytokinesis, but still allow mitosis to continue and remain as healthy cells. So clearly it has something to do with the internal growth factors. Anyway, that's it for this presentation. Again, if you have any questions or concerns or comments about anything that I've covered here, by all means, come and see me during office hours. Otherwise, I will see you next time for the next chapter when we'll be talking about meiosis, the other type of eukaryotic cell division. I'll see you then.